Photosynthesis and cellular respiration. Photosynthesis can be defined as the conversion of light energy and CO2 into carbohydrates for plants to gain energy and continue to live. This process would be considered a redox reaction, meaning that it contains both a reduction and oxidation reaction. The process begins with the oxidation reaction because O2 is created as a byproduct from the photosynthetic electron transport chain's use of an H2O molecule to gain an electron for the process to begin. The reduction reaction part comes from the reduction of CO2 molecules to form carbohydrates in the Calvin cycle for the plants to essentially use as food. Photosynthesis is important because it removes carbon from the atmosphere and converts it into carbohydrates for a plant to sustain life. Photosynthesis takes place in an organelle found in plant cells called the chloroplast. The first major step of photosynthesis is the photosynthetic electron transport chain. This step of the process takes place on the thylakoid membrane of the chloroplast. The electron will move between the lumen and the thylakoid membrane. This chain consists of PS2, PQ, a cytochrome complex, PC, PS1, FD, an NADP plus reductase, and an ATP synthase. It begins by the breaking apart of the molecule H2O into an O2 molecule and a proton. Once the electrons are released, they can start at PS2. The photosystems of this step are meant to charge the electron when its energy is used. The electron begins at PS2 phase where it can be charged by the chlorophyll. The chlorophyll harnesses energy from the sun in order to be able to charge an electron. Now that the charged electron can be carried across the membrane by PQ, where it is then transported by the cytochrome complex until it can be handed over to PC. The electron travels in a Z formation, also known as a Z-scheme. This process of transporting the electron causes it to lose energy, meaning that it must be charged again. The uncharged electron is then moved from PC into PS1, and the light energy harnessed by the chlorophyll can recharge it. The excited electron is then taken by FD into the NAD plus reductase enzyme to be attached to an ADP plus molecule to be reduced into NADPH. While the electron is moving throughout the chain, the protons released in the splitting of water are pumped back and forth across the membrane by the ATP synthase in order to create a gradient to produce ATP. All of the products created by this chain are now in the stroma, with the exception of the O2 molecule, which is released into the atmosphere. The second major step of photosynthesis is the Calvin cycle. The Calvin cycle can also be referred to as the dark cycle because sunlight is not necessary in this step. The energy comes from NADPH and ATP that were previously produced from the photosynthetic electron transport chain. This is the process of carbohydrates being formed from CO2 in three major steps, which include carboxylation, reduction, and regeneration, and it takes place in the stroma of a chloroplast. The first out of the three major steps of the Calvin cycle is carboxylation. This is where CO2 is collected from the atmosphere to bind with a 5-carbon sugar whose name is shortened to RUBP. The major enzyme used in this step is known as ribulose bisphosphate carboxylase oxygenase and can be shortened to Ribisco. Ribisco is responsible for making sure carboxylation occurs properly, but it is also known for causing the reaction to be done slowly. After carbon dioxide and RUBP attach themselves to an active site, the reaction creates a 6-carbon compound. This 6-carbon compound then breaks into two separate 3-PGA carbon molecules. These are the first products of the Calvin cycle. However, O2 can sometimes accidentally be absorbed instead of the CO2 molecule. If O2 reacts with Ribisco, a small complication will occur and the cell will have to find a way to fix it and continue the process. After carboxylation is complete, reduction occurs. The reducing agent of the Calvin cycle is known as NADPH, which was produced from the photosynthetic electron transport chain. The two 3-PGA molecules are reduced by the use of ATP to phosphorylate them, and then two high-energy electrons being transferred to them by NADPH. After all of the energy is transferred, GAP is formed. The GAP molecule and the DHAP molecule combine to form triose phosphates, which are known as the official products of the Calvin cycle. But per 6 triose phosphates produced by the cycle, one must be withdrawn because RUBP cannot be regenerated. The final step of the Calvin cycle is regeneration. ATP is used to give RUBP the energy to regenerate, which means that 3 5-carbon RUBP molecules must be made from 5 3-carbon triose phosphate molecules. In total, 3 ATP molecules and 2 NADPH molecules are needed per CO2 molecule that reacts with the enzyme ribisco. There is an abundance of carbohydrates produced from this step of the process, then the excess amount is stored as starch in the cell. The final step of photosynthesis is photorespiration. This only occurs when oxygen accidentally gets added into the Calvin cycle. The process of photorespiration uses an ATP to properly dispose of the O2 molecule. The enzyme ribisco is also appearing in photorespiration because it begins to combine with O2 instead of CO2. When it combines with O2, it produces one 3-PGA and a phosphoglycolate. It requires more ATP to get rid of the oxygen and adjust the products. Photosynthesis can vary in efficiency because of the plant's location. There are several types of plants in numerous environments, and this could cause a variation in photosynthesis efficiency. As stated by Joseph A. Berry and W. John S. Daunton on page 265 of the Environmental Regulation of Photosynthesis, environmental stress, such as those imposed by drought, salinity, nutrient deficiency, pollutants, or excessively high or low temperatures, have direct effect upon photosynthetic capacity. The next major process is cellular respiration. As stated in section 7.1 of the biology How Life Works by James Morris, Daniel Hartle, and many other authors, cellular respiration is a series of catabolic reactions that convert the energy stored in food molecules such as glucose into ATP. Remember, the catabolic reaction is one that breaks down molecules to create ATP to use later on in the cell. The main catabolic process in cellular respiration are known as glycolysis and the citric acid cycle. Glycolysis is an anaerobic process that takes the glucose or sugars and breaks them down in order to create ATP molecules to use for energy. It produces 2 3-carbon molecules from a 6-carbon sugar or glucose and also creates 2 ATP molecules. The first phase of glycolysis adds 2 phosphate groups to glucose to produce fructose, a 1,6-bisphosphate. This phosphorylated glucose remains in the cell, and after ATP is added to it, it is now prepared with the right amount of energy for the next 2 phases. The second phase can be referred to as the cleavage phase. The 6-carbon molecule is now split into 2 separate 3-carbon molecules, and its isomer dihydroxyacetone phosphate. This phosphate is then converted into glyceraldehyde 3-phosphate. The product of the second phase is 2 3-carbon glyceraldehyde 3-phosphate molecules. The last phase of glycolysis produces 2 pyruvate molecules. These final products of glycolysis will be seen later in order to produce ATP and ADPH, NADH is an electron carrier. The second stage of cellular respiration is the conversion of the pyruvate into acetyl CoA. This part of the process occurs in the matrix of mitochondria in eukaryotic cells. The pyruvate is first transported to this matrix where it can give off CO2 after being oxidized. Electrons from this process are combined with NAD+ to be reduced into NADH, or an electron carrier. The acetyl group of the pyruvate is attached to coenzyme A, or CoA, which will safely transport the acetyl group around the mitochondrial matrix. Now that the pyruvate product from glycolysis is converted into acetyl CoA, it is possible to see the final products of this step that came from one glucose molecule. 2 CO2 molecules, 2 NADH molecules, and 2 acetyl CoA molecules have been formed. The third step of cellular respiration is the citric acid cycle. It can also be referred to as the Krebs cycle or the tricarboxylic acid cycle or the TCA cycle. This step of cellular respiration is important because it's production of high energy electrons, which will later be used in the electron transport chain seen in photosynthesis. The citric acid cycle also occurs in the mitochondrial matrix. It begins by the reaction between a 4-carbon oxalate acetate molecule and a 2-carbon acetyl CoA. This reaction forms a 6-carbon citrate molecule. The final step of the citric acid cycle will regenerate a 4-carbon molecule. The 2-carbon molecules that are released will be released in the form of CO2. While the carbon atoms are being oxidized to create the CO2 products, NAD+ is being reduced to NADH. This cycle is known to produce 3 NADH molecules and 1 FADH2 molecule, which are both electron carriers. These molecules will eventually help synthesize ATP. As stated in section 7.4 of Biology How Life Works, overall, 2 molecules of acetyl CO produced from a single molecule of glucose yields 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. The last step of cellular respiration is oxidative phosphorylation. Now that 2 types of electron carriers have been produced by the previous steps, their energy can be used to create energy molecules of ATP. Electrons will travel through an electron transport chain in the inner mitochondrial membrane to be accepted by oxygen, which will be reduced to water molecules. ATP is synthesized by the transfer of protons and its potential energy, which will be harnessed by the concentration and charge gradient they create. The electron transport chain is embedded in the inner mitochondrial membrane. This chain is composed of 4 protein complexes. Electrons begin their process by entering from either the first or second protein complex, depending on which carrier they were donated by. If they were carried by NADH, they will begin at complex 1, and the electrons carried by FADH2 enter through the second complex. The following steps are considered redox reactions because electrons are being passed through donors and acceptors, which are redox couples, because one will oxidize and the other will reduce the molecules attached to the electrons. Coenzyme Q, or CoQ, transports electrons from the first 2 complexes to the third protein complex. It is reduced to COQH2 when it has accepted an electron. After the electron passes through complex 3, it is transferred to cytochrome C, where it is reduced and diffused in the membrane. This will allow it to travel to the 4th and final complex. Energy is constantly being released from these electrons as it passes through each phase of the electron transport chain. Its energy is used to reduce the carriers in the process or to pump protons back and forth across the membrane. Protons will be stored in the intermembrane space. The potential energy used to create ATP comes from the proton gradient that is created when protons are pumped across the membrane from the mitochondrial matrix to the intermembrane space. This gradient just describes the concentration of protons seen by the reactions occurring. This proton gradient is composed of a chemical gradient and an electrical gradient to create an electrochemical gradient. This chemical gradient refers to concentration levels, while the electrical gradient measures the difference in charges. Protons will have a higher concentration in the intermembrane space and a lower concentration in the mitochondrial space. As the protons try to balance out the concentration, they are stopped by the membrane, which creates a high potential energy in the intermembrane space. An enzyme called ATP synthase is responsible for synthesizing ATP molecules from the energy of this proton gradient created by the electron's movement through the electron transport chain. The potential energy of these protons are released when they travel through a transmembrane protein channel. They travel from the intermembrane of the mitochondria into the matrix, where the movement of protons through the enzyme is coupled with ATP synthase. ATP synthase has two subunits called F0, and F1. The first subunit, F0, creates the channel that the protons pass through. F1 catalyzes the synthesis of ATP from these protons. The proton movement through the channel made by F0 causes the channel to rotate, creating kinetic energy from potential energy. The F1 subunit rotates as well due to F0's rotation. This rotation allows ATP to be created from ADP and an inorganic phosphate molecule. Potential energy has been converted to mechanical rotational energy, which is a kinetic energy, into chemical energy for ATP. After the entire process of cellular respiration, approximately 32 molecules of ATP have been created. This ATP production allows the cell to have a source of energy to do work. Both photosynthesis and cellular respiration are vital for life.